Good morning, and thank you for inviting me to speak this morning. So I'll be talking to you a little bit. Thank you for stealing my joke, by the way, about the blaming on the mother. About kind of a idea that sort of started a few decades ago that was quite provocative at the time, that in utero environment has a profound effect for later in life, 50, 60 decades later, that it will affect the health of the individual. And some of the things that we see that we think of as result of lifestyle as an adult or an adolescent were actually kind of set up much earlier than that. So this is the cover of Newsweek. This is actually quite a while ago, but this is kind of when it started. It made the news for a bit during the news cycle that basically you are set up. You know, anything that's happening to you in your 50s and 60s was actually predicted and set up by your in utero environment. And obviously it's a very sensationalized headline. And bottom line is, yes, but it's complicated, and probably, I think, would be the best way to describe this. So where did this idea originate and how did it kind of come about? So we're going to kind of go back a little bit and do some history. So it all started with David Barker, who was an epidemiologist in England. He worked at University of Southampton. He actually came to Loma Linda and gave a talk. Him and Dr. Longo knew each other over the years. And he came around 2004, I think 2005, to speak here. So he was interested in what were the geographical differences in heart disease and cardiovascular disease in general. So it was heart attacks and strokes that he was looking at in England. And England collected very detailed data on multiple illnesses like cancers, heart disease, just sort of very many categories, and it was all collected at sort of the county level of detail and put together these maps that are actually still accessible today, the current ones, where you can see sort of a health atlas of the country. So he was looking at what areas of the country had the most cardiovascular disease, and they were mostly the poor areas in the northern part of the country at the time. This is late 70s, early 80s. And at the time, it was thought that cardiovascular disease is a consequence of excess, right? So you would expect it that as people are well off, they get excess calories, they don't do as much manual labor, they would become more sedentary, therefore they would get more heart attacks and strokes. And actually it wasn't the case, it was mostly in poor areas. And he started looking at other maps that had, sort of other conditions, and he noticed that the map for infant mortality and the map for birth weight overlaid almost perfectly to this, but it wasn't a one-to-one at the same time. It was maps 50 to 60 years earlier. So the same area 56 years earlier. So these are the kind of what the maps look like. This is the most recent one that looks at heart disease, still mostly in the northern part of the country. And then low birth weight. Now, they don't overlap right now because you're missing that sort of delay. So, and low birth weight today is very different than low birth weight. We're talking people that were born in the 1920s. Low birth weight in the 1920s was malnutrition, crowding, infectious disease, no access to medical care. Today, low birth weight is much more likely to be things like hypertension, vascular disease, diabetes, and other things like that. So he sort of made this observation that it was not really, the dogma at the time was that when you're rich and you have a lot of calories, you will eat a lot, you'll get big, and you'll get heart disease. It seemed that it was actually kind of a reverse. So he looked at other conditions and noticed that it was the same thing for type 2 diabetes, for high cholesterolemia, for all the things that sort of make that metabolic syndrome, syndrome X, all those sort of categories were there. So he started presenting this data, he published it, and he actually got some fairly pointed critiques that you're missing a lot of confounding factors. This could be a case of care is not as good in those areas. This could be poverty, this could be substance abuse, this could be, anytime you're looking at population level data, there's a lot of things that you don't see, and you're aggregating a lot of things that may not have a common thing, and association does not lead to causation, and you have to be careful. So there was a lot of debate, but people looked at other populations, and then they started, so this is the data he was looking at, kind of put in a graph. So these are 1921 to 1925, and this is infant mortality plotted against coronary heart disease 50, 60 years later. So both in men and women, the more an area, the higher the infant mortality in an area, the higher the coronary artery disease was 50 to 60 years later. So that was kind of his initial observation. So he started overlapping these, found a really good correlation between the two. And they were looking at 1968 to 78, and then for people that were born between 1921 and 24. That was their initial data cohort. So cardiovascular disease, diabetes kind of seem to go hand in hand. And so they said, okay, let's take away the limitation of look at an entire population and see if we can follow individuals. The good thing is a lot of these people tended not to move. They lived in the same area their whole life. They had accurate birth records, and they had accurate hospital records, and they could be followed throughout their life. So they started doing follow-up studies. So they looked at adult hypertension, coronary artery disease, diabetes, and they also looked at the weight that they had when they were 1 year old. So it wasn't just birth weight, is how they did in that first year of life. And the other thing that they looked at carefully was there is small and there is small. Whenever I have a patient that has a baby that's growth restricted, technically, if they're less than 10th percentile, it sort of raises alarm bells and we start to wonder why and what's going on. And there's sort of a phenotype that you have when it's a fetus that is not getting adequate nutrition through the placenta. They tend to be what we call asymmetrical IGR, where the belly tends to be very skinny. They tend to redistribute blood flow, and they will have, they preserve the brain and sacrifice the trunk and the carcass, basically the liver, abdomen. So there'll be a skinny, a thin baby with a sort of relatively large head. And that, you can see that when we sort of measure the ponderal index. And some babies are just small because somebody has to fill out the bell curve, right? Like somebody has to be short, somebody has to be small. There's small mothers, small fathers will have small babies, and that's not necessarily a sign that there's a problem. So looking at the ponderal index, they were sort of able to pick out these kids that were the sort of asymmetric IUGR, and that correlated really well also with it. So, especially with hypertension. So these are kind of the kids. You see the one on your left, I guess, is sort of the skinny one. There's a big baby on the right, and kind of the middle is more one that might be small, but is actually proportionally small. So kind of all different sizes, and it's sort of captured in those body measurements. And they were able to get those from the birth records because they measured length and, head size and that kind of thing. So they said, let's look at individuals rather than look at a population because we may be missing things. So this was a study done in one area. There were 370 men. They were at age 64, and they went back and looked at their birth weight and saw that the smaller they were, their odds ratio of getting type 2 diabetes skyrocketed. And this was adjusted for BMI because the critique was always, well, what happens if, somebody, whatever, doesn't matter what they're born, if they get very obese as an adult, they might get diabetes anyway. So that effect persisted. So even in people who were not necessarily overweight, they still got diabetes at a much higher rate. So it sort of supported that. So then people started looking, and there was literally an explosion of studies looking at this. So as you can see, things like Indian children, in Indians in South America, all over the world, Africa, European populations, Danish, even the nurses' health study, really the same effect was seen over and over in multiple population at sort of the epidemiological level. So it seemed to be not some kind of unique thing that was linked to some other, effect in the population. It seemed to actually kind of be a universal thing. So people started looking, how can we sort of put together a mechanism that would explain this? Why is this happening? Why would we be seeing this? So Barger came up with this thing that he called the Thrifty phenotype hypothesis. So his hypothesis was that the in utero environment basically sets up our sort of insulin resistance glucose metabolism thermostat. So if you are in utero and you are at a deficit and you're not getting enough nutrition, you'll be much more likely to be sort of glucose sensitive later in life. And this is sort of shown in this diagram. So fetal malnutrition, people looked also at various components of the diet. It seemed to be specifically amino acid deficits. So it's protein deficit that really causes this effect. Simply replacing it with calories from like sugar or something does not abolish this. So in some populations, they've seen even calories are okay, but the protein is deficient, you'll still see this effect. So the idea was that because of the malnutrition, somehow there's less beta cell mass in the pancreas, there's decreased fetal growth, the fetus is extremely sensitive to insulin as far as its growth. That's how, babies of diabetic mothers end up very, very big. They metabolize glucose very well, which ends up then spilling over that often if the mother does not have good nutrition, the infant will also not have good nutrition. So that sort of first year of life is critical, which then you end up with dysfunctional adult B cells that were kind of set up in utero, and non-insulin dependent diabetes, hypertension, and all those other things. So the other thing that ends up happening too with as they follow these kids is a lot of these small kids end up catching up and actually overshooting the growth chart. So they'll be born, they'll be small. It's both a biological thing where they will do this kind of spontaneously, but also they'll get extra care, right? My baby is too small. We need to feed the baby. It's always like, it's the greatest crime a grandmother can have to have a grandchild, right? That is too small. Like you're gonna put extra effort in to get that kid fed. So when they look at the growth curve of these kids, they actually catch up in sort of in the first year of life, and actually often will overshoot and then stay high and stay above. They tend to be very sort of, when they're exposed to calories, they tend to sort of metabolize it and put it down as fat. The idea is that they were told in utero, expect a harsh environment where food will be scarce when you are born, and be very, very efficient at holding on to calories when you are exposed to them. So their thermostat was set as that, as you expect to not have a lot of calories, but then they had plenty of calories, and then it sort of kind of triggers this downstream effect. So it's sort of shown here that maternal nutritional supply, fetal nutritional demand, that balance ends up in a fetal adaptation. And if postnatally it's a match, then you have normal adult risk. If the postnatal demand is not equal to the supply, generally the postnatal supply ends up being higher, and you have a background of having been exposed to a low caloric environment in utero, then your risk of disease will be much higher. That's sort of the working hypothesis of this. So people said, do we have any historical evidence of this? What other places can we look for evidence for this? And most logical place to look at is there were populations that were exposed to famine, extreme caloric restriction, extreme protein restriction. And so people went back and looked, and there was one that was not that far in history at that point, which was the Dutch famine. So the Dutch famine was sort of, if somebody had designed a study to look at how undernutrition and pregnancy would affect fetuses and the adults later on, they would have done a really good job. This was sort of the perfect storm. It was November 1944 to May 1945, so it was that last winter of the war. It occurred in Holland because there was initially a lack of supplies coming in because the trains coming into Holland had been A, sabotaged, and B, there was a strike where the workers refused to help the Germans run the trains. So the initial part of the famine was actually a lack of transportation in the middle of winter to bring in supplies. When the Germans restored the trains, they decided that they were going to punish the population by still not bringing in supplies. So it was very abrupt. There were daily rations given out that were recorded, so we know how much calories people got. There was very good record keeping. At some points where it got really bad, the rations were as low as like 600 calories a day. And there was an effort to give children and pregnant women slightly higher calorie levels, but still there was just no food available. So once the war ended, the nutrition was restored very quickly to normal levels. So it was sort of a very well-defined time. And there were a lot of pregnant women, and there were some women too who became pregnant during that period of time. So, and on top of it, all of these newborns were sort of enrolled in the healthcare system in Holland and had very good medical records that were kept for life because they were seen in sort of a, sort of state hospitals, clinics, etc. And particularly the males had very good medical records because at the age of 18, every single male has a comprehensive physical exam, psychiatric evaluation for the compulsory military service. So every single boy that was born during that time has this really detailed physical and a psychiatric eval at age of 18. So this cohort sort of was able to be followed through and looked at. So this was kind of the timing and why it was kind of interesting. I'm going to try to make sure, here we go, good. So the boxes are the 3 trimesters of pregnancy, right? So you had a group that the mother was already pregnant and was already entering the 3rd trimester as the famine started. So it was a late exposure. They were only exposed during the 3rd trimester. They delivered during the famine. So then the newborn was also exposed for a few months. There were some that got pregnant not long before the famine started. So they were exposed in the mid-trimester and then delivered after. And there were some that got pregnant like at the tail end of it, and so were exposed in the 1st trimester only. So within that cohort, you had selective exposure by trimester, which was really helpful because it turns out that it does make a big difference to what trimester you're actually pregnant when you're exposed to this stress. So many epidemiologists descended upon Holland and started looking at this and seeing what kind of effects this had for these as they became adults. So this is I have a summary that's much shorter, but just to kind of show you, kind of what kind of data came out of this. So plasma glucose was higher, plasma insulin was higher, but as you can see, it kind of depends what trimester you're exposed. BMI was actually not significantly different, so it's not that they became obese. They had metabolic issues despite having a similar weight. And it sort of varied by which trimester you're exposed to. The hypertension here was sort of continuous, and they actually calculated because they saw what the rations were and were able to calculate that. And some people got less or more protein, that it's 0.8 millimeters per 1% decrease in your protein to carbohydrate ratio. So they sort of were able to see very precisely what was going on. Even obstructive airway disease was higher, and sort of perceived poor health, it seemed to be that if you're exposed in the first trimester, it was that. And there was also a fair amount of early pregnancy losses, a lot of miscarriages in this cohort. So kind of in summation, if you're exposed in late gestation, mostly you had glucose intolerance. So it was mostly metabolic issues that you had. Mid-gestation, you also had metabolic issues, some renal problems, obstructive airway disease. Early gestation, you got a lot of hits. Obesity seemed to actually only affect women, but even altered coagulation, lipid profile, coronary heart disease. And so here under stress sensitivity, there was actually studies done where they looked at rates of psychiatric disorders, and there was a higher rate of schizophrenia, and just kind of overall bipolar disorder. There were not just physical sequelae of this, but psychiatric ones as well. So that was very useful, sort of more data that this was a thing. And it still don't have a mechanism. We have some better ideas that because it's so reproducible, it seems to have different effects. There's even a dose-dependent effect on some things, that it's a real thing. So people looked at other historical famines. There were publications on all of them, but they are nowhere near as detailed or as good as the Dutch famine ones. The siege of Leningrad lasted for 3 years, so most of those were throughout the entire pregnancy and even after. The other difference in Leningrad was that it was an impoverished, malnourished population before the siege happened. It just got worse during the siege, and it didn't really get better afterwards. So there was no sort of time effect of saying you were only exposed to caloric deficit during this time. It was a continuous thing, so harder to isolate. There was China during the kind of revolutionary years. There was an entire province. Not as good, also follow-up data, not as good birth weight data as the other ones, so harder to follow. And then with war in Nigeria was also studied fairly extensively. The only thing was that there was no birth weight data available at all because these were not, majority were not delivered in the hospital. So it was sort of limited, but all of these kind of showed a similar effect. So this is kind of an updated, after they looked at all these things and did a lot of sort of detailed metabolic studies on these patients, they sort of came up with an updated Thrifty phenotype hypothesis where both maternal malnutrition, and actually it turns out that maternal hyperglycemia too, because they looked at current cohorts, like mothers who are diabetics and that kind of thing, can actually lead to fetal and infant malnutrition because we were talking about that sort of asymmetric IUGR. You can have all the nutrition you want, If the placenta is not working properly because you have vascular disease or something happened during early development, you are going to end up with a malnourished fetus because the transport is not happening properly, the baby's not growing well. So it's not just malnutrition, it's sort of anything that causes the sort of placental interface to not work properly can trigger this. And then you have sort of global changes in growth, metabolism, vasculature, anything from muscle, liver, the HPA axis with insulin resistance people have looked at. That's where kind of the stress response comes in, where these people tend to be much more sensitive to stress. They don't tolerate, stress as well. They have worse responses to it, and it kind of ties in also with a psychiatric illness that was noted in the population. So great, we have this great, awesome hypothesis, and how are you going to test it? In humans, it's hard. It's very hard to get, biopsies. It's very hard to control exactly what people eat. It's hard to wait 60 years for these things to happen. So not the ideal epidemiological subject, but not the ideal, sort of, if you're looking for a mechanism. So sort of the limitations the way I talked about before, anytime you do population studies, you're going to have confounding factors. You're going to have things like, variability in access to care. You're going to have variability in substance abuse. You're going to have genetic backgrounds that are different. So it's hard to sort of tease out those kind of things. You have things like, some people live next to a chemical dump site. If you live in Flint, Michigan versus you live somewhere else, you're going to have differences that are not due to your birth weight. Birth weight might be a surrogate for something else that happened to you during pregnancy, but is not, sort of a clean study. So that's kind of the point. Small for gestational age is often an endpoint of multiple things, which are all kind of hard to tease out. And so many times when patients even come in with a small baby, we do all the labs, we do the ultrasound, we try to figure out, and we say, we don't know why your baby is small. It just is. And it might happen to you again, it may not, but, we don't have a good reason. You don't have even any risk factors, that kind of thing. So what's the mechanism? So the natural place to go is animal studies. So that's some of the work that I did with Dr. Longo. When I came into his lab, he worked with hypoxia and at high altitude sheep, and he was interested in what happens to the fetus that does not have enough oxygen. He had even done also some of the early political work. His early work was on carbon monoxide in the placenta, and he had testified in front of the EPA and was kind of part of the initial smog rules and that kind of thing. He testified the effects on pregnant mothers. So he was really interested in that, and he had looked at the placentas from high altitude and noticed they were very different morphologically, and said, let's look for a mechanism. So we looked at the mice because sheep are bigger, more expensive, longer gestation. That was kind of his animal choice. So we picked this strain of VBNJ. The bottom line, it's very easy to breed. It's very docile. It's very fertile. It's very inbred, which is good because you're removing your sort of genetic variability out of the pool. And it's also the parent strain for a lot of genomically modified strains. So a lot of knockouts and things kind of come out of the strain because they're such good breeders. So we figured if we ever want to do any sort of knockout studies, this would be a good one to have as our sort of background strain. So we actually did first the hypoxia experiment. So we put them on half an atmosphere. The gestation in the mouse lasts around 18, 19 weeks. So it's essentially the last sort of 2 days of the pregnancy. And I kind of found this out by trial and error. If you put them longer than that, they get really sick, they get really stressed out, and they will actually eat their litter. They get too stressed out. They feel like it's too stressful an environment. They will just, sort of to protect themselves from when they get really stressed, they think there's predators around something else, they will not breed well. So kind of last part of gestation, we did hypoxia. And then because of all the protein restriction data, the protein seemed to be the one that mattered. We actually fed them an isocaloric diet that had half the protein that normally does versus regular protein. We collected the placenta and then various organs from the pups right after they were born. So these were not adults that, sort of started showing this. We wanted to know what sort of the initial effect. And then we initially looked in the placenta, and we used microarray technology to look and compare hypoxic placentas versus normoxic versus protein restricted. And this is sort of what the placentas look like. That's kind of what struck initially. That's a control, that's a hypoxic. If you do actually, when you measure the morphometry, there's a lot more vessels in here. And the density is higher, the vessel walls are thinner, and they're much more tortuous when you kind of go in at higher field. And we thought, oh, I bet it's going to be something to do with vascular genes, VEGF, some other growth factor that has to do with blood vessel growth, angiogenic factors. And so we did induce an effect. The protein deprivation, they actually had smaller weight, so they were growth restricted. And then we looked at the different genes on here. I have a summary slide, so you don't have to read every single. I hate when speakers do this, but I still haven't gotten out of the habit of putting these up. But there was no, trust me, there's no genes here related to blood vessel development. So we didn't see anything vascular. But we did see some epigenetic regulators, so things that had to do with DNA methylation or histone acetylation. So we'll talk about that, what that means in just a minute. And then same thing in hypoxia, a lot of things that had to do with blood, like there was some angiogenesis, but it had more to do with erythropoiesis, so more RBCs, which makes sense, and also a lot of genes that have to do with epigenetic changes. So we started looking initially for vascular things, and then we kept seeing these genes that had to do with epigenetics. So we actually kind of went backwards because we saw this, and then we went back and said, what is this about? And then kind of walked backwards through the Barker hypothesis and said, oh, this might be a mechanism, because you look here, there's different things that they do, but the one thing that they have in common is control of cell death, which makes sense in types of stress, and DNA methylation. I should probably add here histone acetylation. So what is epigenetics? So it literally means something on top of the genome. So you have the sort of classic cascade of gene goes to protein, and gets expressed and has an effect. But the gene expression is actually controlled at multiple levels, one of them being how tightly packed the DNA is. The DNA is not a big string, it's actually kind of rolled into these very organized chromosomes, and the parts that are tightly wound are not accessible for proteins to read the gene that's on them. So when it's packed with a chromatin, it's not accessible, those genes can not become available, and the cell, when needed, will unpack the chromatin, will alter its structure so it doesn't bind to the DNA and release areas that then various genes can be expressed. So that allows each cell to be different, and then different times in the cell to express different genes, change its phenotype, but also it allows sort of certain areas of the cell to be permanently more or less available to be read. And the other mechanism is DNA methylation, where there's actual methyl groups that go on top of the DNA, and I have another slide that shows that a little bit better, where if the area of the gene promoter, so the part of the gene that's before the gene starts, where all the transcription regulation happens, if it's methylated, it can't bind to it. So it physically blocks it from binding and reading a gene, so it's a way for the cell to silence a gene, and a way to turn it on is to remove those methyl groups. So we thought that's kind of a nice way to change your phenotype and, sort of in the face of external cues, and change the way your genes are expressed that don't have to do with the genes you inherited. It has to do with also your environment. So we decided to look, because we saw hypertension was one of the very common sort of phenotypes, we decided to look at the renin angiotensin system. And I hadn't looked at this for a very long time. And it turns out I still have PTSD from medical school, so I'll just go through very quickly. It's angiotensinogen. Renin turns into angiotensin 1. ACE turns it into angiotensin 2, and it does a million things at the vessel level. So it controls hypertension. It's very sort of a central system in the hypertension regulation, and it's known to be controlled through epigenetic mechanisms, how tuned up or down this is, so we thought that was a good place to look. So trust me here, basically we looked at the brain RAS, at the systemic RAS, which would be kind of the liver, and there were differences in multiple places in the expression level of both the receptors, renin, angiotensinogen, and also the ACE enzymes were differentially expressed in the protein-restricted mice. So we thought, great, it's there. Let's see if there's anything going on with the promoter. Do we actually see any DNA methylation changes? So this is what this looks like. So start codon, RNA transcription starts, you get your exons that will get put together, and then you have these CPG islands, which are areas that have CG pairs, which can be methylated. And the more methyl groups you put on it, the less likely it is to get transcribed. You can actually fully silence it if the whole area is methylated. So you see this in cancer a lot, where normally silenced genes lose their methylation and all of a sudden turn on that were only developmentally expressed. Those were supposed to be expressed during in embryonic development, all of a sudden turn on because they lose methylation. So we looked at the promoter of one of the genes in the pathway, and I have a better one too, but basically each dot represents a methylation sites, and there were differences between protein restricted and non-protein restricted, and here you can see it much more clearly, where in this particular gene, which is one of the receptors, protein restriction basically demethylated it substantially. So it turned the gene on for the ACE receptor. And so this is actually the actual locations where it's on. So there we found the proteins are different, the RNAs are different. We even looked at some of the miRNAs which control gene expression at sort of another level, and those were also different. And then we looked at the hypoxic ones. We also saw a lot of changes to ACE. It was not quite as clean. It was a little more confusing because there were some that could be putatively good, some of them that seemed neutral, and some of them definitely seemed like sort of a problem. And there was not as much DNA methylation changes, but there were changes in these miRNAs, which are small RNAs that control other RNA expression. So we did see, and they're also considered epigenetic because their levels are controlled sort of adaptively. So that kind of put the story together that in the animals we actually found there was a mechanistic thing, and it's not what we're looking for. It's just one of those things where we started doing an experiment and it kind of took us in a completely different direction. But it's since been repeated by many other groups. They've shown changes in DNA methylation for genes that control metabolism in particular. And some of the proteins that are actually targets of anti-diabetic drugs seem to be some of the receptors are methylated or methylated in these kids. So there's more and more evidence that that's probably the mechanism. So the idea is you get exposed to stress in utero, your body says, I'm going to be born in a very stressful, food-restricted environment. You prepare yourself for that by methylating your genome appropriately, and then you're born and it turns out that you actually have plenty of food around, and you're sort of maladapted to your environment because of it, more likely to get metabolic disorders. So we kind of went from epidemiological data, we went back through historical data that supported that, and then through animal studies that show that this does work and there is a mechanism for it. And it sort of show that maternal nutrition is, as we kind of, programs that enhance maternal health and maternal nutrition will have cascading effects 50, 60 years down the line. It's not just about getting that pregnancy or getting a healthy newborn. It's actually setting up that newborn for success later on. People have looked at various interventions for this. It appears actually that folic acid seems to work for this. In the animal models, they were given protein-restricted diets and high levels of folate, and it reversed a lot of these effects, and a few other methyl donors. But a well-balanced, sort of nutrition will have sort of profound effects downstream at the population level. And that's it. Thank you.
Well, we have a few minutes here for questions. And of course, the only dumb question is the one you didn't ask. So Dr. Gheorghe will be right here to answer some of those questions. And could we pick up another mic for here? And so we have a nice little roving mic that's going to be passed around. Here's one right here. And it's not pickleball, but we'll throw it to you. And then you can actually speak to it. But this is the beginning of a theme that you'll be hearing throughout this day regarding the disease of obesity. So there's a lot of bias around this disease, and we wanted to start out the morning strongly with the emphasis of how even the in utero effects bring this to place. So could we see the first question? And again, there we go. There's a good question up there. Can we get it up to that gentleman right up there? And all of you, if we run out of time, because we will stay on time, but if we run out of time, be sure and note those questions because they're very, very critical to this conference. Go ahead.
Hi, Dr. Gheorghe. Thank you for that presentation this morning. It made me wish I remembered more of my biochemistry from medical school. But I guess a couple of mechanistic questions. Well, why did you put the mice under hypoxia? Was that to simulate perhaps a mother with cardiovascular disease or with low perfusion of the placenta? Yeah,
that was sort of actually the primary model that we had with the hypoxic sheep because you reliably induce a placental insufficiency like phenotype. So they're smaller. They have lower oxygen. They have a lot of the adaptation in, mothers that have like vascular disease that have placental insufficiency.
Okay, and then quickly, the second question was, one of the main mechanisms seemed to be upregulation of the angiotensin converting enzyme 2, which from medical school I remember that played a significant role in hypertension. But to me, it doesn't seem to explain, the diabetes increase risk and also the cardiovascular increase risk. So it seems to be still insufficient in explaining everything.
Correct. So there are other groups that have looked at the diabetes pathway, like PPR gamma and a few of the other transcription factors and proteins involved in that. Some of the diabetes insulin receptors. And those are also altered. We looked only at the vascular side because we were a vascular lab, so we were plumbing only. So.
Question right over here.
Have you, or are you aware of others that have looked at high protein diets rather than protein deficient, the very popular ketogenic diet that many put themselves on today? And if so, what would be the effects of that diet?
Not aware of any animal studies. There's some sort of anecdotal data of people who are sort of very avid ketoers who became pregnant. We discourage it clinically, but I have to say I haven't looked very closely, so I don't want to misspeak, but I haven't seen anything. I see someone lifting their hand, so I don't know if it's related. But I I don't think it's been looked at very much as far as I know, but I could be wrong and I just sort of missed it. So,
as a doctor in nutrition, I'm going to have to correct that. The ketogenic diet is not high protein. It's a low protein, high fat. So just to kind of correct that. Other questions here?
At the end, you mentioned taking folic acid and that it would reverse. And could you just expound on what you were saying there? What is it actually that, we think it's neural tube defects. So what's the benefit in relationship to obesity?
it seems to be methyl donors seem to reverse it. So folic acid can do it. It sort of goes through the carbon, small carbon metabolism, and like L-arginine and a few other amino acids. But they're basically thought of basically diets that are high in methyl donor groups for some reason seem to reverse it. That's what people have been looking at.
How long do
Those were animal studies, so they were the same sort of protein restriction we did, except that they supplemented them with folic acid or methyl donor groups and reversed the effect.
It doesn't matter what trimester is what I'm saying.
These were throughout, for those animal studies, I think they were throughout the gestation, so they had put them on the diet before. Sometimes switching their diet in the middle of pregnancy for the rodents especially, they get really stressed out and miscarry, have all kinds of complications, so you kind of have to commit early.
Any other questions? Here's one right here. Just a moment here. Let's get this on the mic.
If you want an excellent book on epigenetics, a book called The Epigenetic Revolution by Nessa Carey, I think really gives a complimentary view to your talk. I really enjoyed it. Thank you.
Thank you.
All right. Well, thank you all very, very much. Thank you, Ciprian.